Good morning, everybody. My name is EthanJoshuaNOng, and I'm here to present to you all my research entitled The Vaping Alternative Evidence of E-Cigarette Oncogenicity in Vitro and Comparison with Effects Induced by Cigarette Smoke. So, traditional cigarettes are known to have about 70 cancer-causing components. There are about 1.1 billion smokers in the world today. And of course, these 1.1 billion smokers, about 8 million of them die each year. Now, moving on to e-cigarette, its smoke delivery system is relying on a vaporizer, which heats the vape juice within a cartridge and converts it to smoke. E-cigarettes are very accessible to a large population of minors, and 1 out of 10 high school students use or have used the vape. It is projected that there will be 55 million users by the year 2021. Currently, the vast amount of research conducted, millions know about the carcinogenic effects of traditional cigarettes and have been straying away from them in favor of supposedly safer e-cigarettes. Now, e-cigarettes are relatively new to the cigarette industry, and there hasn't been nearly as much research on them as there has been on traditional cigarettes. With that said, people are hailing the e-cigarette as the safer option, but is it really? We barely know anything about its carcinogenic properties or biological repercussions, so how can we say that it's safer? So a breakdown of the two types of cigarettes show their very stark differences. We can see that both have a very addictive substance, which is nicotine, but other than that, the two vary significantly. E-cigarettes have ultrafine heavy metals present, while cigarettes have the poisonous arsenic and cadmium. These stark contrasts lead me to believe that perhaps e-cigarettes are not as safe as people proclaim them to be. And with that said, the objectives of my study are to determine if the effects of e-cigarettes are on different cancer hallmarks in comparison with cigarette smoke extract, and to determine if effects of e-cigarettes are limited to toxicity or structural injury to cells and not oncogenicity. So the cell line I used was A549 human alveolar basal epithelial cells. These are adenocarcinomic, and this is the primary cell line I used for all my assays and staining experiments. The e-cigarette I used is called the Orion DNA Go, and it has a nicotine concentration of 50 milligrams per ml, and this is on the higher side as nicotine concentration goes. I used a 50 ml syringe to extract the smoke, and I exposed the DMEM with 10% FBS, which is my media, to 100 hits of this e-cigarette, with one hit basically being one pump of the syringe, so one up and down movement. So moving on to my methodology proper, this is the orientation of my syringe and e-cigarette, which allowed me to extract the smoke and infuse it with the media. I then cultured A549 cells in normal DMEM with 10% FBS first, and after they were confluent, I infused the media with the e-cigarette smoke in varying concentrations. I called this e-cigarette smoke infused media, or e-cigarette smoke extract media, or ECSE for short. I then proceeded to do 4 main experiments, check for actin cytoskeletal remodeling, migration rate, cytotoxicity, and cellular stress through reactive oxidative species. So the first one I will be talking about with you all today is cytotoxicity through the LDH assay. The LDH cytotoxicity assay is a colorimetric assay that relies on detecting the enzyme lactate dehydrogenase, or LDH. This LDH is indicative of cellular damage as a result of increasing cytotoxicity. Now, based on the graph, we can see that e-cigarettes have a high cytotoxic effect at high concentrations, with the 1X in particular being highly cytotoxic. Do note that traditional cigarettes are also known to cause high levels of cytotoxicity. And next, I did the DCFDA staining to monitor the reactive oxidative species, or ROS levels. Now, DCFDA is a fluorogenic dye that allows me to measure the ROS activity of a cell, and superoxide, which is the red molecule here, is the precursor for most cellular ROS. The DCFDA staining results are quite telling, and we can see that the oxidative stress experienced by a cell is directly proportional to the concentration of e-cigarette smoke. Note that each jump in concentration brings significantly higher oxidative stress than the previous one. This is also similar to the effects caused by traditional cigarettes. Afterwards, I wanted to check on the structures of my cells, and I decided to perform actin cytoskeletal staining. The cytoskeletal staining involved using Alexafluor 488 phalloidin, and I used this because it has a particularly high affinity for F-actin, or to F-actin, and has a high photostability as well. The media I used was normal media, which is my DNO 10% FBS at first. Then I changed it to media with e-cigarette smoke in the concentrations of 1x, 0.5x, 0.1x, 0.01x, and my control, which is 0x. So the first notable structure change we see is an increase in filopodia density. Filopodia are slender bundled actin, which a cell would normally use for migration, adhesion, or cell-to-cell communication. This increase in density is usually linked to cancer and metastasis because filopodia are known to stimulate cell migration as well as scan the topography of the ECM, or extracellular matrix, to check for possible binding sites. So these are indicated by the orange arrows here. Next, we see tunneling nanotubes, which are indicated by the yellow arrows. And these are made of actin and are usually used for intracellular connections, short or long range. So cells use TNTs to exchange components such as organelles, proteins, and nutrients, and these TNTs are normally associated with cell invasiveness, and the cells can also transfer mitochondria between each other, and this is known to be correlated with drug resistance. Next, we see the presence of peripheral ruffles, and these are actin-based veil-shaped protrusions that can be found in the periphery of the cell. These peripheral ruffles eventually become circular dorsal ruffles, or CDRs, and these CDRs support cancer cell migration by softening the cytoskeleton through the disruption of stress fibers. Both peripheral ruffles and CDR are also linked to the tumor cell nutrient uptake through micropinocytosis. So the peripheral ruffles are indicated by the red bracket over there. And now that we also see that the cells are actually shrinking and their respective nuclei are migrating towards the periphery, this suggests that the cell is getting ready for migration or movement because the cytoskeleton is disorganizing itself. This is clearly indicative of a migratory phenotype. We also see multilobulated nuclei here within some cells, and these are nuclei that aren't typically round or spherical in shape. This is associated with cells arrested or delayed in S phase of the cell cycle, and these cells could potentially become cancerous. This delay in the S phase, or at rest, or on the other hand, may be attributed to possible DNA damage or replicative stress, and multilobulated nuclei are commonplace among cancer cells. And then we also saw multinucleation, and this is the presence of more than one nuclei and is implicated with tumor formation and drug resistance. An increase in tumorigenicity may lead to tumor relapse, possibly metastasis. So, what are the other notable structures here we see? We also see invadopodia, which is indicated by the pink arrow, and this is known to degrade ECM, or the extracellular matrix, and is linked to metastasis. The red arrow here shows a thick peripheral actin rim around a round cell, and this is a sign of motility because lamellipodia tend to form as a result of this thick peripheral actin, and lamellipodia is linked to motility. The white arrow shows a fan-shaped leading edge, which is a finger-like actin protrusion, and this is known to stimulate cellular migration. So moving on to the additional cichlids, we see the same structures I mentioned earlier, such as invadopodia, thick peripheral actin, and philopodia. So here we also see multilobulation, and then here we see tunneling nanotubes or TNTs and some multinucleated cells. And in this picture, which is the highest concentration of cigarette smoke, traditional cigarette smoke, we see a lot of tunneling nanotubes and multinucleated cells. So based on the pictures I showed, both traditional cigarettes and e-cigarettes cause significant structural changes to the cells. We see the same changes as I listed here, and the main difference when it comes to these changes is that the effects of e-cigarettes are on a less dramatic scale. If you remember, we saw much more TNTs or multilobulated nuclei or multinucleated cells as compared to the cells exposed to e-cigarettes. So what e-cigarettes did not do, what e-cigarettes, however, rather did, was to shrink the cell, and the traditional cigarettes did not shrink the cell whatsoever. So this may indicate that the e-cigarette cells are actually more migratory or metastatic. So I wanted to check this migratory potential. I did this by conducting the wound healing assay. I cultured A549 cells exposed to different concentrations of e-cigarette smoke in a 96-well plate until confluency, and I scratched and waited 14 hours to view them. So these are the results, and they show that there aren't any significant changes to the migratory rate of the cells aside from those exposed to the highest concentrations of e-cigarette smoke, wherein those cells may become significantly slower relative to the control. This decrease in migratory rate may be attributed to the high cellular toxicity at this concentration. Now, comparing the results of e-cigarettes to traditional cigarettes show that the latter actually makes the cell move faster, and that the migration rate drops off at the highest concentration, which is similar to what we see for e-cigarettes. In summary, the two types of cigarettes do induce significant structural changes to the cell, but the e-cigarettes do so on a less dramatic scale. The traditional cigarettes increase the migration rates of cells, while e-cigarettes have no significant effect on this migration rate. Both show the decrease in cellular migration rate at high cigarette smoke concentration, and this may be linked to the high levels of cytotoxicity at this concentration. Overall, the e-cigarettes were deemed to be highly cytotoxic, especially at high concentrations, and they also induced high oxidative stress, as seen from the ROS levels, and they induced significant structural changes, and they had no effect on cellular migration. In the future, I plan to do these experiments with a normal lung cell line instead of A549, which is adenocarcinomic. I also plan to check how e-cigarettes could potentially regulate SCAL1, which is known to be upregulated by smoke, and then check the effects. Thank you for listening.